So I'm very happy now to introduce our second speaker for today, which is Professor Rafael Maldonado. I see he has joined already, so he's ready to start, I guess. So Professor Maldonado is the founder of the Laboratory of Neuropharmacology at Pompeu Fabra University. His work focuses on the neurochemical and anatomical mechanisms of drug dependence, particularly opioids, cannabinoids, and nicotine. And today he will share his findings on the role of gut microbiota in food addiction, exploring how gut bacteria influence neurobiological processes and identifying potential biomarkers and innovative treatment strategies. So once again, we'll have time for questions in the end. And please join me in welcoming Professor Maldonado.
Thank you very much, Elena. Thank you very much, Nasia, for the invitation, for this kind invitation.
Thank you for joining us.
I will share my screen. Okay, I guess that you are now seeing my screen.
Yes.
Okay, so as Elena told you, I'm going to talk about the involvement of the gut-brain axis in food addiction. So these are two very polemic issues, gut-brain axis, food addiction. So what we know about the involvement of the gut, a peripheral organ, in the function of our central nervous system, the knowledge is, well, several decades ago, it was already identified that cells of the gut are able to release some hormones that will modify, will influence our behavior, a centrally mediated response. And the first one, of course, was cholecystokinin, CCK. But later, GLP-1, peptide YY, a lot of hormones that are released. And at the beginning, it was identified that all these gut hormones were able to control our feeding behavior. That was the starting point, feeding behavior. But later, we start to understand that another important mechanism is also involved, and is the gut microbiota. Gut microbiota are all the microorganisms that we have in our gut. Of course, a lot of bacteria, but not only bacteria, viruses, fungi, a lot of microorganisms. And how these microorganisms that are outside of our body, let's say, just in the gut, how they are able to control our responses in the central nervous system. The classical mechanism, first one that was identified, was the metabolic pathway. All these microorganisms are able to release several metabolites, several chemical substances, like a small chain fatty acid, that cross the gut barrier, go to the systemic circulation, and they are able to pass the blood-brain barrier. So these metabolites released by the microbiota, finally, they are going to arrive to get the central nervous system. We are talking about small chain fatty acid, but cytokines, even neurotransmitters, like they are able to release all these substances that finally get to the central nervous system. We also have another important mechanism, that is the immune response. These microorganisms will be identified on a external tissue, and we are going to generate an immune response. Immune response that will produce several substances like cytokines, TNF-alpha, interleukins, that you know very well that these substances are able to modify our behavior. And they are able to cross the gut barrier, they are able to cross the blood-brain barrier, some of them. And this is the second mechanism. Another mechanism are the autonomical enteric nerves. All this gut is innervated by autonomical nerves that can directly modify the response in the central nervous system through compounds that are released by this microorganism. For example, oxytocin. This microorganism, in the previous talk, you hear about oxytocin. Oxytocin is released by our microorganisms in the gut, and this oxytocin through the vagus nerve modify behavior in the central nervous system. This microorganism release GABA, a neurotransmitter that modify also the responses through the central nervous system. So you can see a lot of tools that the microorganism have to modify central responses. Another one is the neuroendocrine pathway, because there is a link, a direct bidirectional link between the HPA axis and the gut. The release of corticoids will modify our microbiota will modify the release of corticoids. So these are a lot of bidirectional interactions between brain and gut. Well, another important issue is when we take our microbiota, of course, depend on our diet. When we take a high nutrient diet, we are going to increase the thickness of the gut barrier. However, if we take the classical fast food, a lot of carbohydrate, a lot of fat, we are going to decrease this gut, the thickness of this gut barrier, and to produce a lot of interaction that we don't want to produce. Okay, so you can see here the large amount of mechanisms that we have to control this influence of the gut microbiota on the brain function. Of course, gut microbiota is modified by a lot of peripheral factors, environmental factors. The first one is the diet, we already mentioned. Factors like the pollution of the environment. If you live in the countryside, you are not going to have the same microbiota that we living in the cities. The drugs, classical example, if you take an antibiotic, the physical exercise, all these are going to modify our gut microbiota. So this gut microbiota is a very good link between environmental factors and brain. And as I said, the first behavioral response that was demonstrated to be controlled in some way by the gut microbiota is food intake. But later, we have a lot of responses, cognitive responses, stress, social interaction, fear. And now I'm going to present data about food addiction. But let me previously introduce a little bit the models that we have to evaluate, the animal models that we have to evaluate in animals this gut-brain axis. The first one are the germ-free models. Animals that don't have any microorganism in the body. And I anticipate these germ-free animals have dramatic alterations. Alteration in neurogenesis, dendritic growth, microglia, neurotransmitters, synaptic plasticity, and behavior, of course. And that's just because we suppress all the microorganisms in these mice. But other models are the use of antibiotics. We have models using antibiotic in mice. Fecal microbiota transplant, I'm going to talk a little bit about that. Prebiotics. Prebiotics are substances that are going to modify our microorganisms in the gut. And the probiotic is the administration of these microorganisms directly by oral route. So these are the classical ones. Of course, there are others, but these are the classical ones. And let me introduce a little bit this one. The fecal microbiota transplant. Because previously to these studies on food addiction, we have some interesting data with this procedure of fecal microbiota transplant. For instance, this article that we recently published in Cell Metabolism. It's a good journal, as you know. So what we did here, our clinicians just evaluate patients with obesity, and they identify in these patients with obesity, comparing, of course, with non-obese individuals, they identify a particular microbiota signature that is responsible on some metabolic responses, and is also very well correlated with alterations in the hippocampus, alterations in the volume of the hippocampus. And what we did, we took the microbiota of these subjects and we transplant by oral route, the fecal microbiota, we transplant by oral route to our animals. Of course, we gave previously antibiotic to avoid the previous microbiota. And what we observed was that when we transplant the microbiota for non-obese individuals, the memory of the short-term memory of our mice was better. And if we transplant from obese individual, we impair the short-term memory. So we transplant the phenotype of humans, let's say, to animals, just with the gut microbiota. Another example, this paper that we published also recently in Gut, where we demonstrate that the inhibitory control, we did the same with the inhibitory control in subjects that have obesity. You know, inhibitory control is very important to inhibit our instinctive responsive. That's crucial for addiction. If we don't have inhibition, we are going to become addict very quickly. Eh? And in humans, a classical test, I'm not going into details, you want later with the questions, a classical test is the Stroop, the test of Stroop, and just measure impulsivity. And in our animals, I'm not going into details now, but we also measure impulsivity and cognitive flexibility in operant behavioral model. And interesting, we obtained the same. We were able to transplant the impulsivity phenotype in animals, just taking the gut microbiota of humans, of obese or non-obese humans. So these are examples, very recent examples, where we are able to modify the behavior of our mice, just taking the gut microbiota of humans. And these animals mimic the behavior of humans in some way. Inhibitory control, cognitive deficit. So now what I'm going to present are the most recent data that we have in this involvement of the gut-brain axis in food addiction. Of course, food addiction, you know, or probably you know the polemic on this issue. Food addiction, it exists. Well, first, just remember the control of food intake. Eh? Food intake is crucial for survival. So we have two different mechanisms, parallel mechanisms, just to promote, to control food intake. The first one is the homeostatic mechanisms. If energy is lacking, we are going to increase food intake. If we have a lot of energy, we are going to promote satiety. And this is controlled by a lot of hormones in the periphery, some of them in the intestine, other in the pancreas, in the adipose tissue, that inform us about the amount of energy that we have. And this information goes to the hypothalamus, and the hypothalamus just promotes satiety or hunger. It's very simple. But you know that, well, we are not in a natural environment, but imagine a natural environment. In a natural environment, sometimes we find the food, other times we don't, because whatever, roughness, whatever. So we need to accumulate energy for the period where we don't find food. It's for that reason that we have another parallel system to control food intake, that is the allostatic control. The allostatic control is that we are going to eat just because food produces pleasure. It activates our reward system. And that's the reason, because food is going to release dopamine in the nucleus accumbens. And you know that it's directly related to food intake, to reward, sorry. So we control with the hypothalamus the amount of energy that we have. And we have another system that says, take food because that produces pleasure. And you go to a bakery and you found the chocolate, and immediately you go inside, even if you have taken your lunch, just because you want to have the reward. However, there is also an inhibitory control in this reward. That is the prefrontal cortex. You know that the prefrontal cortex is the highest hierarchic structure for the control of our behavior, and is the responsible of providing all the rational control in our behavior. And this is the inhibitory role of the cortex. So for that reasons, we have a rational control of our food intake. But take in mind that we have both systems in parallel, homeostatic, allostatic control. Well, this allostatic control reminds a lot the mechanisms of drug addiction. Because, well, let's see, before to go to the mechanism, the food addiction. Food addiction is certainly a very polemic issue. It was already defined in the 50s, that means 70 years ago, by Randolph. And what Randolph says, what Randolph hypothesized, is that some particular individuals with some particular food develop changes in the brain that mimic the changes occurring during drug addiction. And basically, the main issue of food addiction is that we have an individual vulnerability. Some individuals develop food addiction, others not. We have some particular foods that are able to produce, to develop this behavior, the palatable and obesogenic foods, and these processes are related, this food addiction is related with other eating disorder, obesity, binge eating. It's a close relationship. And of course, there are a lot of similarities with drug addiction. Look, well, DSM-5, as you probably know, doesn't include food addiction in this manual of diagnosis. And that is something that probably you know. DSM-5 includes substance use disorder, does not include food addiction. However, there is a scale, that is the Yale scale, that is widely used in clinic to evaluate food addiction, and that mimic this substance use disorder criteria to define drug addiction. So we have this tool to diagnose food addiction in humans. And you're going to see that is a tool that we have used in our studies, Yale Food Addiction Scale. And the Yale Food Addiction Scale, they use the criteria of substance use disorder. So if we summarize, I'm not going to explain the 11 criteria of substance use disorder, but if you summarize, basically we can describe, we can say that the hallmarks of addiction are persistence in seeking the drug. You cannot stop to seek the drug. Motivation. The drug is really the center of your life. Your life is really focused on looking the drug, seeking the drug, nothing else. It's really the center of your life. And compulsivity. Compulsivity means that you are going to take the drug in spite that you know that the drug is going to have negative effects on you. You take the drug in spite of these negative effects. And of course, there is the craving, the necessity of having the drug. But with this criteria, there is this Yale Food Addiction Scale. And in terms of mechanism, of course, in 30 seconds, the mechanism that we could explain during 3 hours. But just the key idea is that all drugs of abuse start the addictive process because all of them are able to activate the dopaminergic pathway from the ventral tegmental area to the limbic system, particularly to the nucleus accumbens. And this is the common issue in starting addiction to any drug, whatever the drug is. All of them are able to release dopamine in the nucleus accumbens. And also, you know, this bidirectional interaction between limbic system and nucleus accumbens. Also, Nora Volkov has demonstrated that in addiction, there is a dramatic decrease in the activity of the prefrontal cortex areas. You can see here on the left the brain, or the activity of the brain in a healthy volunteer, and in the right, the activity of the brain in a cocaine addict. You see the arrow, you can see that the prefrontal cortex activity is dramatically decreased. That's happened with cocaine addict. That's happened with heroin addict, with cannabis addict, with alcohol addict, in the case of ludopathy, in addictive processes not mediated by drugs. So every single addictive process has this decrease in the activity of the medial prefrontal cortex area. So in this bidirectional control, the prefrontal cortex is losing. You are going to have a response that is just mediated by the limbic system, not by the prefrontal cortex. And you are going to take the drug in spite of any negative consequence. Well, so you see the similarities between these mechanisms involved in drug addiction and the allostatic control of food intake are involved exactly the same brain structure. Nucleus accumbens, limbic system, ventral tegmental area, and cortical areas. Well, some few years ago, it was postulated that this gut microbiota probably has a role in food addiction. It was postulated this hypothesis. You take a diet that is unhealthy, you are going to modify your gut microbiota, you are going to modify your behavior due to this gut microbiota, and you are going to increase the intake of unhealthy food. It's a vicious circle. That was postulated. And in our study, we want to evaluate this hypothesis. So first, before evaluating this hypothesis, we need to understand a little bit more about food addiction. So in this previous article we published in 2020 in Nature Communication, we identify the specific neurobiological substrate, one specific neurobiological substrate in food addiction. There was this Nature Communication paper. So, of course, this was already mentioned in the previous presentation. We need models, animal models with a translational value. If not, it doesn't mean to do research in animals. So how may we develop a model with translational value in animals, a model of food addiction? It was difficult, eh? It was difficult, but I get that we got it. Eh? I go just to present the positive results. Eh? So if we need to have variability because some individuals are going to develop addiction, other not, use outbred mice, of course, not use inbred mice, use outbred. So it was what we use in these first studies. We compare a lot of different kinds of food, only positive results. Standard food with regard to chocolate flavor pellets. Chocolate flavor pellets, what does mean? That means that the caloric value of this pellet is exactly the same than the standard pellets. We avoid the bias of the calories and of course the bias of caloric of food restriction. Our animals have food exposure in normal conditions, and these are not at all animals with food restriction. It's a normal animal with normal food, and we go to an operant model to evaluate the capability of this animal to seek for this kind of food. I guess that you know an operant model. No, Elena, they know or not? Do you think that it was that I explained a little bit?
I think you can explain the model because also it's very small on here.
Okay, so an operant model. An operant model means that the animal is going to associate a non-conditioned stimuli with a conditioned stimuli. That's me. Non-conditioned stimuli, taking food, taking a food that I like. Conditioned stimuli, a lever. The animal must to press an active lever, non-conditioned stimuli, to obtain palatable food, the conditioned stimulus. And the animal learns very quickly, believe me, animal just learn very quickly to associate the pellet with the administration of palatable food. So we start to train the animals during 5 days in affective ratio 1. That's me. The animal press once the active lever and obtain a very small pellet, a pellet of 20 milligrams that the animal just eat, doesn't produce ascites, very small pellet. Then we move to fixed ratio 5. That's me. The animal must press 5 times in the lever in order to obtain 1 pellet. Look the training. The training is very hard. We train the animals during 115 days. Extremely long experiment. The life of a mouse is 2 years and a half. So this corresponds to a very long exposure to this training. And we evaluate exactly the same 3 criteria of DSM-5. Persistent to response, motivation, resistant to punishment, compulsivity. How we can evaluate this in mice? So, persistent to response. Each operant session has a duration of 60 minutes. We have 25 minutes active period, then 10 minutes of pellet-free period. That means the animal, the light is off in order that the animal understand that this is a pellet-free period, and whatever the animal does is not going to get the pellet. So the animal during these 10 minutes, if it's pushing the active lever, is not going to obtain anything at all. And later, we put the light on, and the animal again push the active lever to obtain the reward. So animals that have a persistent is going to push even if he knows that he's not going to get any reward. This is persistence. Motivation, progressive ratio scale. What does that mean? That means that to obtain the first pellet, the animal needs to press the active lever once. Second pellet, 5 times. Third pellet, 12 times. Fourth pellet, 21 times. Later, 33, blah, 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 until 5,500 times. For no one is going to press 5,000 times the active lever to get the reward. But imagine the model and imagine that this is real a quantification of the motivation, because an animal press 75 times to get a single pellet, whereas another press just 12 times. You have a breaking point, the point that the animal says, stop, I don't make any additional effort because it doesn't worth to get just one single pellet. We evaluate this motivation. Compulsivity, resistant to punishment. Fitness ratio, 5. The animal press 1, 2, 3, 4 times. When the animal press the 4th time, gets a foot shock. The animal doesn't like the foot shock. Press the 5th time, the animal gets another foot shock, and later the pellet. That's compulsive. The animal press is a compulsive animal. Need to get 2 foot shock in order to get 1 single pellet. That's compulsivity. So we evaluate this. I'm not going to the details to don't be very long, but we evaluate these 3 criteria and using a percentile. An animal that go over the percentile have a positive criteria, whereas other animals that don't go through the percentile don't have the positive criteria. So you can imagine that each animal is going to have 0, 1, 2, or 3 criteria, positive criteria. Look the classification. We evaluate this compulsive behavior in a very early period and at the end of the training. Persistence to response. Animals that were trained with standard food, all the animals control very well the persistence. They stopped at the moment that the light was off. In the early period, the animals that are trained to obtain chocolate always stop. However, in the late period, we obtain what we want to obtain in this case. That was a very high variability. That was the mean of our experiment, to obtain this variability. That means some animals that develop this persistence, whereas other animals don't. And look, for instance, this animal. This animal press almost 600 times during this period. 10 minutes period, that means the animal press once every second during 10 minutes, looking for the pellet. Persistence is enormous. Look the motivation. Motivation, standard food, they don't have a very high motivation, but look with chocolate, even in the early period, there is an animal with a very high motivation. But look the variability and the motivation of the animal for chocolate in the late period. These 2 animals almost press 2,000 times to obtain 1 pellet. Can you imagine a mouse pushing in a pellet 1, 2, 3 until almost 2,000 just to get 1 single pellet? That's extremely high motivation. And resistant to punishment, also saying there was 1 outlier here. Sometimes you have outliers with the standard food, but look with chocolate in the late period. There are animals that got more than 40 chokes. That's mean, imagine the compulsivity of this animal. The animal doesn't care about the receiving choke or not. It's just looking for food, for palatable food. So we have this criteria, and we have each animal has, imagine this animal that correspond to this and this. This animal has 3 criteria. Imagine that this animal will correspond to this and this. This animal has only 1 criteria. Okay? So we have from 0 to 3 criteria. And animals that have 0 criteria, we consider resilient to develop the disorder, food addiction. Animal that has 2 or 3 criteria, we consider addict animals that develop food addiction. In the standard food, no one developed food addiction, 0%. Whereas 70% have zero criteria. So this is the classical response with standard food. With chocolate flavored pellets, 22% develop food addiction. 55, the majority were resilient. So this 25 remembers a little bit the percentage of addict in the case of exposure or some particular drug of abuse. So we consider this model has a translational value. Later we can discuss about that if you want, no? But we consider that it has. So we want first to, in this nature communication, to use this model to evaluate the pathway. And we use a procedure that is called DREAD, the signer receptors exclusively activated by the signer drug. I'm going to very briefly summarize this procedure. It's a chemogenetic inhibition or activation of a pathway. What we do? We are going to introduce in the cells that we want a particular receptor with a viral vector. So we administer this viral vector, you can imagine in the brain area that we want, and when we administer this viral vector, the viral vector will recombine with a particular sequence. In this case, the sequence is CRE. CRE is an artificial sequence that we can introduce in the animal. So the animal, imagine a mutant, a transgenic mouse that expresses CRE in the glutamatergic neurons. That was the case, the experiment that we are going to present now. If the animal has CRE in the glutamatergic neurons and we inject in the medial prefrontal cortex our viral vector, the animal is going to express this viral vector only in glutamatergic neurons in the medial prefrontal cortex. So this viral vector contains a receptor that can be excitatory or inhibitory. In our case, we use, the result that I'm going to present is with inhibitory receptors. And this receptor is going to be activated by a molecule that we administer, clozapine N oxide. So look the tooling, we have our receptor that we have administered in the medial prefrontal cortex. As the animal has CRE only in the glutamatergic neuron, this receptor is going to be expressed only in the glutamatergic neuron of the medial prefrontal cortex, and at the moment that we administer this compound, we are going to inhibit the receptor, to inhibit the pathway. So when we administer in the medial prefrontal cortex, during a period, this was during a period of 4 weeks. Why? Because our hypothesis is that if we inhibit the medial prefrontal cortex, we are going to remove the inhibitory control, and the animal will develop addiction, is our hypothesis, will develop food addiction. So we inhibit all the projections of the medial prefrontal cortex during 4 weeks. And we train the animals during 4 weeks in this paradigm. Of course, we do the electrophysiological studies to verify that indeed we have inhibited this glutamatergic pathway. I'm not going into detail, but believe me, we have inhibited this pathway, as we demonstrate with this electrophysiological study. And then we are going to see the behavior. What's happened with this animal when we inhibit these pathways, this glutamatergic pathway from the medial prefrontal cortex to other areas? Well, what we observe, the behavior, just the pressing for the palatable food was not modified. But look, the motivation and the compulsivity was increased. And if we consider animals addict or not addict, though the control, 15% were added. Very few animals, yeah, because it's a very short period. It's only 4 weeks. So only 15% of the animal. But when you inhibit the glutamatergic projection from the medial prefrontal cortex, 42% of the animals become addicted. So these pathways are indeed involved in the development of food addiction. However, look at what we did. We inhibit all the pathways. So of course, this experiment is not selective. And the referees of Nature Communication says, please let me know which one of these pathways is involved. That's a little bit more complicated, but we did it. How we did? We inject in the nucleus accumbens a retrovirus, containing CRE. So it's a retrovirus. We inject in the nucleus accumbens. So CRE, this sequence, is going to all the neurons that project to the nucleus accumbens. Hippocampus, ventral tegmental area, amygdala, prefrontal cortex. However, we inject our dread vector only in the medial prefrontal cortex. So the receptor will be expressed only in the medial prefrontal cortex cells that contain CRE. That means the glutamatergic projection only to one pathway, to one particular site, that is the nucleus accumbens. We selectively inhibit the pathway from the medial prefrontal cortex to the nucleus accumbens using this retro approach. Of course, electrophysiology study demonstrate that we inhibit this pathway, and the animal respond more or less similarly. And look here, we have an increase in persistence to response and increase in impulsivity. 50% of the animals become addicted in this very short period. So this first study demonstrates that the mouse vulnerable to food addiction can be generated. Well, this I didn't show, but that there is a medial prefrontal cortex nucleus accumbens pathway that is crucial in the development of food addiction. We identify the pathway. So I'm going to finish with two studies that we have published in Journal of Clinical Investigation, and this one was just now published in Gut from British Medical Journal. It's a very good journal. So we want to know if this microbiota is involved in this food addiction, using this model. And we also want to know what is the link between the link between this gut microbiota and the brain in this particular response. So we focus only positive result, because if not, we are not going to finish. We focus on an epigenetic factor that is microRNA. So we evaluate microbiota and microRNA in these animals. MicroRNA was published in Journal of Clinical Investigation, microbiota in gut. So it was submitted, is already published. So we do exactly the same protocol, 100 days, training the animals for food addiction, and we evaluate the criteria of addiction and other phenotypic traits: impulsivity, aversive learning, compulsive learning, a lot of 4 different behaviors that I'm not going to detail, just to have a full characterization of our animals, to really be sure that our animals are addicted and that we can extrapolate the result to humans. So that allows us to classify animals as vulnerable to develop addiction or resilient to develop addiction. And you can imagine that we take these animals, vulnerable and resilient, to study the gut microbiota signature and to study the microRNAs. MicroRNAs we study in the medial prefrontal cortex, as you can imagine. And of course, I'm not microbiota in the feces. So we have here the experiment. So standard food, no one develop addiction. These normal animals develop 25% develop addiction. So a little bit more than in the previous experiment, but it's more or less the same. Eh? We replicate the previous experiment, of course, with different results, as you can imagine. 20% become addicted. So we select the extreme animals. Is that for that reason we had the 3 criteria and the 4 phenotypic trait, really the extreme. And we analyze microbiota and microRNAs. Let's see microbiota. Microbiota, we have a change. I put this here because there is we have a change in the abundance of several genus of microbiota. Enteroradus, decreasing Enteroradus, Lanospiracea, Lobaculum, Blautia. We have a very, very nice decrease. And increasing anaeroplasma. Anaeroplasma is a bacteria classically related to a positive effect, to a negative effect, to a negative effect. And this is increase in the addict. Whereas Blautia and Enterorhabdus are the classical positive bacteria for our health, let's say. So there is an enhancement in negative bacteria and decrease in positive bacteria in the food addict group. This is in the genus. If we go to the phylum, phylogenetically, you see that the genus is down. Genus, family, and on the up, very up, you have the phylum. So these are very large groups of bacteria. Just to have a general view. With the phylum, of course, you don't know anything at all because it's very high in the phylogenetic scale. But you have a general idea that allows you to make the correlations. That was the reason because we went to the phylum, just to make the correlation. And look, Bacteroidales, you have a decrease, phylum Bacteroidales, you have a decrease in the addicted mice. And you have an enhancement in Prevotellacea. So you have, if you make the ratio between Bacteroidales and Prevotellacea, negative and positive, you observe a dramatic decrease in the addictive. So this is a very nice criteria to correlate with other parameters, in particular with the microRNA. Keep this in mind, because now we are going to the microRNA. Later we are going to the microRNA with this ratio. So in humans, of course, we want to extrapolate to humans. Our collaborators, our endocrinologists, took a cohort of humans that has food addiction and others that don't have. Just to look the microRNAs, plasmatic microRNA, you can imagine, and microbiota. So look at the human, they show, we do the opposite translation. Usually you go from the human to the animal, here we go from the animal to the human. Because in the human, we evaluate exactly the same than in animals. We made the scale of persistent motivation compulsivity that has very good correlation with the food addiction, with the Yale scale of food addiction that was used for this criteria. And when we go through the microbiota, we observe a very good correlation with food addiction with some particular genus, but one of them just we pay attention, that was Blautia. This beneficial bacteria was dramatically decreased also in humans and in animals. So we use this probiotic, we use Blautia, just to see what's happening if we administer Blautia to our mice. Just because we have this data in animals and in humans, demonstrating the dramatic decrease in Blautia in addiction. And of course, we did other experiments with prebiotics that I don't have time to show, but just let's see the probiotic. The probiotic, if we administer Blautia by oral route, so animals are colonized by Blautia, the behavior to obtain chocolate is not modified. Animals press exactly the same. However, look the development of addiction. There is a dramatic decrease in motivation. There is a dramatic decrease in compulsivity. No one of the animals that were exposed to this probiotic Blautia developed food addiction. In this experiment, 21%. of the control animals develop food addiction, more or less the same numbers than before. Remember, 22, 25, here 21. But the really important is 0% develop food addiction with this probiotic. We have similar result with prebiotics, but I don't have time to show. So microRNAs, we observe in the medial prefrontal cortex of our mice a decrease in a large number of microRNAs. And in humans, there were some common changes in plasmatic microRNAs in food added in humans and microRNAs in the medial prefrontal cortex of animals. Plasmatic human, medial prefrontal cortex in animals. And also, I think I will not. Also, this correlation was shown in male mice, in man. Okay, so a correlation also in sex, gender. In male mice and in men, there was a decrease in microRNA 29C in the medial prefrontal cortex in the plasma, and also a decrease in microRNA 665. So we focus in these two microRNAs. If is decrease in animals in the medial prefrontal cortex and in humans that have food addiction, probably they have a role in this addiction, and probably they are correlated with the changes in microbiota. Look the correlations. For the correlation, let's go through this ratio. Remember this ratio between the phylum, Bacteroidalis Prevotellacea. There was a very nice ratio that interestingly correlates with microRNA 665, microRNA 29C. So look the correlation here. Correlation between, well, similar results in plasma in humans, in medial prefrontal cortex in animals, very good correlations with the changes occurring in the gut microbiota. So let's go through the possible role of these microRNAs in food addiction. I finished with that. What we did? Remember that we have a decrease in the medial prefrontal cortex in microRNA, in these microRNAs, in animals that develop food addiction. So try to mimic this decrease in microRNA in the medial prefrontal cortex. For that, we administer, I'm not going to the detail, but we administer a tag decoy in the medial prefrontal cortex. This tag decoy is going to recombine with the microRNAs, in particular, this tag decoy with the microRNA 29C, and this tag decoy is going to decrease the expression of microRNA 29C selectively in the medial prefrontal cortex. Remember that was one of the changes in animal addicts. Okay, so we expect to develop food addiction, so that makes everything more easy because we don't need to use the long protocol of 4 months. We just use a protocol of 4 weeks because we try to develop addiction in a short period by decreasing these microRNAs. And indeed, I'm only presenting the positive result. There was a lot of negative result with all the microRNAs. But look, there is a very interesting enhancement in persistence and in motivation. Compulsivity was the same. That means from 15% of animals added in the control group, we got 50% animals added when we decrease the expression of this microRNA. And with the other microRNA, with the microRNA 665, we did the same. We decrease the expression in the medial prefrontal cortex, and we're taking a very different result because persistent motivation was not modified, but look compulsivity. Compulsivity was dramatically decreased, was significantly enhanced. And in terms of addiction, 36% of the animals become added over to 1,6. So you see that microRNA 29C is involved in persistence and motivation. MicroRNA 665 is involved in compulsivity. Each one of the microRNA seems to be involved in one particular hallmark of addiction. So, in conclusion, the medial prefrontal cortex nucleus accumbens pathway is crucial for the loss of eating control. The loss of eating control is associated with specific microbiota and microRNA changes that are closely related both in humans and in animals. So our animal model has a translational value, at least in the case of these experiments. And several of these specific microRNAs and microbiota play a crucial role in the development of food addiction, and may represent this microRNA, may represent the link between the gut microbiota and food addiction. This can be the link. So the result, why they are interesting? First, because this plasmatic microRNA and this microbiota could represent biomarkers of food addiction. And second, the probiotics and the prebiotics could be evaluated as possible therapeutic approaches for food addiction. Could be evaluated. We are far away of the clinical application, but that's open the possibility to evaluate these prebiotics and probiotics in food addiction. Well, and of course, you can imagine this is a work. This work was not only from my group, but in my group, I will underline the role of Laura Domingo and Solveiga, and the role of Elena Martin. Has been the three person more directly involved in this research. Of course, to thank all the endocrinologists from the University of Girona, led by Professor José Manuel Fernández Real, the group of Bruckerman in the University of Barcelona, the group of Aurelius Buroca in the University of Vilnius, and the group of Raúl Antero in the University Autónoma of Barcelona. Thank you very much.
Thank you very much, Rafael. This was a fascinating presentation, I have to say. Very clear, I thought. So if we move on to the questions, we already have a couple questions that were asked by spectators. So one that came quite early on was by Catalina. She's asking, given the current understanding of the brain's reward system, do you think addiction can be fully cured, or is it a lifelong management issue?
Well, addiction is a chronic disorder. So addiction must be, we must think that we must take care of our patient during the whole life, let's say. I make the similitude of addiction, hypertension, and diabetes, for instance. Imagine that you have hypertension, you have diabetes. You must take care of you during your whole life, but you're going to have a completely normal life. Normal life, avoiding, in the case of hypertension, taking your medication and avoiding the salt. In the case of diabetes, taking your insulin or your medication and avoiding the rapid sugars. But your life will be exactly the same. And in the end, it will be the same. Avoid the circumstances that may promote the restatement, stress, exposure to the drug, exposure to the condition stimuli. The patient can have a normal life in spite of this chronic disorder.
Thank you. I agree. And then we had also another question by Mihaela, a bit more specific. She's asking if referring to food addiction and given the rising use of GLP-1 receptor agonists like Saxenda, Ozempic, etc., in the management of food addiction and obesity, whether you might know about their long-term efficacy in modulating neural pathways associated with reward and hunger, and what are the most possible significant neurological or psychological or effects, I guess, and how might these impact the broader adoption of these medications in treating food addiction?
Of course, GLP-1 compounds have not been yet evaluated in food addiction. Remember that these compounds were introduced for type 2 diabetes and now for obesity. Are extremely promising, that's for sure. Are extremely promising. But the link, you are going to, with the GLP-1, you are going to have first, the effect on food intake, second, the effect on metabolism. So I will first focus for GLP-1 on, let's say, the peripheral response, decrease of food intake, decrease in fat content, improvement on the triglyceride, on the cholesterol. This is the insulin resistance. This is the everything that has been demonstrated. The future, I really think that GLP-1 can have a major role in all the I'm now speculating. So we are until now everything was the reality. Now the speculation. I speculate that with GLP-1 we are going to have a promising target for everything that is related to the inhibitory control of food intake, because there is a clear involvement in the rewarding process. So I hope that with the GLP-1 compounds, we could be able in a future to study the specific role, the specific utility of these compounds for treating cheating, food addiction, this disorder. But I mean, I'm speculating. Of course, remember one of the main side effects, the price of this compound.
Thank you very much. And if I have time to ask myself one quick question, I know there's a debate when it comes to food addiction on whether it's a chemical addiction or a behavioral addiction. I was curious what your take on this position would be.
I support the fact that these are behavioral addiction. A behavioral addiction because, but I mean, this is my opinion, because you know that there is people that say food addiction, eating addiction. There is an alteration, there is a modification in the personality. You compare people that has are positive in the Yale food addiction scale and people that are not, you find a particular personality range, particular personality characteristics in this human with food addiction. So there is a clear vulnerability to addiction. And of course, you're going to become addicted to palatable food. But at the moment that this food is something that you need for life, I will not go to chemical addiction in this case, in spite of the similar mechanisms, even your personality traits, similar changes.
Okay, I understand. Thank you very much. That was a very great presentation. Thank you for this.
Thank you.
And I will just quickly reply to Rafael, who is asking if it will be possible to watch back all the presentations. All the presentations here are recorded and they will be available in at least a couple weeks on YouTube. So for now, it would be best to watch them live, obviously, to ask questions and everything, but they will be available in the future as well.
Okay. Thank you very much, Elena and Nasira.
Thank you very much.
Thank you,
And
Professor.
goodbye.